in this video, we're going to cover cellular respiration. We're going to break down both aerobic and anaerobic processes. Let's get started. First of all, what is cellular respiration and why is it important? There's no point in going through the steps of cellular respiration and trying to memorize them if we can't see the overview picture and understand the beauty of this process. Cells in living organisms are like chemical factories, performing millions of reactions, and these reactions help the cell survive, grow, and reproduce. Now, in order for these reactions to occur, cells require energy and a source of atoms. We require energy to function. Energy is important to all metabolic processes. That's the first thing you need to know. Our mission here, our goal is to produce energy. Okay? We'll come back to this overview diagram. Now, food is an external source of energy. The energy contained within its organic molecules is derived from the sun. If you recall, photosynthesis produces oxygen, as well as organic molecules that are used by the mitochondria of eukaryotes, for example, plants and animals. What's it used for? Fuel, for cellular respiration. Respiration breaks this fuel down using oxygen to make ATP, energy, because remember, our mission here is to produce energy. The other products, carbon dioxide and water, are used in photosynthesis. All right? Look at this. How can you not fall in love with this process, the beauty behind these processes? That's respiration and how it connects with photosynthesis. Let's go through one more thing. The term that's used to describe the sum total of all the chemical reactions carried out by an organism is called metabolism. And we can divide metabolism into two categories, catabolism and anabolism. Catabolic pathways break down complex molecules into simpler and smaller units, and we release energy. Whereas anabolic pathways build complex molecules from simpler molecules, and we need an input of energy. With that said, cellular respiration is a catabolic process because we're breaking down an organic fuel. Now, we have three sources of organic fuel: glucose from carbohydrates, fatty acids from fats, and amino acids from proteins. But we're going to be breaking down sugar to show the steps of cellular respiration because most cells use glucose as their immediate source of energy. So here's our equation. We'll come back to this, okay? Cellular respiration includes both aerobic and anaerobic processes, with and without oxygen. But the term cellular respiration is often used to refer to the aerobic process, okay? Just keep that in mind, and we're going to break down both. Now, to truly understand how glucose and other organic fuels yield energy, we need to look at the transfer of electrons during chemical reactions. You will see how the transfer of electrons plays out. It's incredible. These electron transfers are called oxidation-reduction reactions, or simply redox reactions. The loss of electrons from one substance is called oxidation, and the addition of electrons to another substance is called reduction. The reason why it's called reduction is because we're adding negatively charged electrons to an atom, and as a result, this reduces the amount of positive charge on that atom. All right? Substances that lose electrons are oxidized, and substances that accept electrons are reduced. Let's draw this out. We have substance X that's going to transfer its electrons to substance Y. So this substance X is the electron donor and is called the reducing agent. It's reducing Y. Substance X is oxidized because it loses an electron here, because it's the electron donor. And substance Y is the oxidizing agent because it accepts the electron. It oxidizes X by removing its electron. Okay, applying this to our cellular respiration equation, if we look at our equation here, glucose becomes oxidized and oxygen becomes reduced. Okay, why are we going through this? Because it's important to understand that the more reduced a molecule is, the more energy it has. And you're going to see this play out because attaining electrons is important in harnessing energy. Keep that in mind. Let's move forward. Cellular respiration is broken down in a series of steps. We can divide it into three metabolic stages: glycolysis, pyruvate oxidation and the citric acid cycle, and oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. Electrons are being removed from the glucose and are stored in the electron carriers NAD+ and FAD. You'll see how energy is transferred in the making of NADH and FADH2, and how during the final stage of cellular respiration, they pass their electrons and are converted back to NAD+ and FAD. And the energy released is used in ATP production. Okay, let's now subtract complexity and go through the first metabolic stage, glycolysis. Glycolysis occurs in the cytosol of the cell. First, glucose is transported into the cell via GLUT transporters or glucose transporters. It occurs in 10 steps and can be divided into 2 phases. We have the preparatory phase or the energy investment phase. This is steps 1 to 5. We have to invest some energy to get some back, inputs and outputs. Then in the payoff phase, steps 6 to 10, we're harvesting energy in the form of NADH and ATP. Let's break this down further. Starting with glucose. Now, glucose contains 6 carbon atoms, okay? This is carbon 1, 2, 3, 4, 5, and 6. We're going to add a phosphate group to the 6 carbon of glucose. We're going to phosphorylate glucose and it becomes glucose 6-phosphate. The reason for this is so that glucose can't leave the cell because the plasma membrane doesn't have transporters for phosphorylated sugars. We're cuffing the glucose so it can't leave. This step requires ATP. So ATP becomes ADP. ATP is the phosphoryl donor in this reaction, and this reaction is catalyzed by hexokinase. Then we're going to convert glucose 6-phosphate to fructose 6-phosphate. This is a reversible isomerization reaction. We're going to move the carbonyl group to carbon 2, and the enzyme catalyzing this reaction is phosphorhexose isomerase. From here, fructose 6-phosphate is transformed to fructose 1,6-bis-phosphate. This is an irreversible reaction. We're going to invest some ATP in this step, and phosphorfructokinase 1, or PFK1, is the enzyme catalyzing this step. So PFK1 is going to transfer the ATP to fructose 1,6-bis-phosphate. Recall that in the previous step, we moved the carbonyl group to carbon 2, so that now carbon 1 is free to be phosphorylated. So the reason why it's called bisphosphate is that we have 2 phosphate groups at 2 different positions. We have a phosphate group at carbon 1 and carbon 6. Moving on to step 4. Fructose 1,6-bisphosphate splits to form these 2 carbon sugars, dihydroxyacetone phosphate or DHAP and glyceraldehyde 3-phosphate are formed. This is catalyzed by the enzyme ATPase. So as you can see here, we're splitting fructose 1,6-bisphosphate. Now when we get to this step, glyceraldehyde 3-phosphate is the only one that can actually continue through glycolysis. So what we're going to do is we're going to convert dihydroxyacetone phosphate, or DHAP, into glyceraldehyde 3-phosphate by triose phosphate isomerase, and this reaction is reversible. As a result, we have 2 glyceraldehyde 3-phosphate, and that is the preparatory phase. Steps 1 to 5, we invested 2 ATP and produced 2 glyceraldehyde 3-phosphate because glyceraldehyde 3-phosphate is the only one that can continue through glycolysis. Let's continue to the payoff phase, starting with glyceraldehyde 3-phosphate. Remember that we made 2 glyceraldehyde 3-phosphate, so each step in the payoff phase happens twice. Glyceraldehyde 3-phosphate is being oxidized to form 1,3-bisphosphoglycerate. This step is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. When you hear dehydrogenase is involved, this is a hint that NAD+ is involved. And so in this step, okay, NAD+ is reduced to NADH. Recall that NADH plays a role in ATP production. It's an important electron carrier. Electrons are being transferred to NAD+, which we'll get to later in the lecture. From here, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. This is a reversible reaction. The enzyme catalyzing this step is phosphoroglycerate kinase. It's going to transfer the phosphate, the phosphate group from 1,3-bisphosphoglycerate to ADP. And so we're going to produce 1 ATP. This is known as a substrate level phosphorylation. because we're producing some ATP by direct transfer of a phosphate group to ADP, substrate level phosphorylation, and we've produced ATP. Then 3-phosphoroglycerate is converted to 2-phosphoroglycerate, where the phosphate group is transferred from carbon 3 to carbon 2 by the enzyme phosphoroglycerate mutase. Okay, 2-phosphoroglycerate then becomes phosphorenol pyruvate, PEP for short, in a dehydration reaction. We're going to be removing a water molecule, and the enzyme catalyzing this step is enolase. Finally, in the last step, PEP is converted to pyruvate. Pyruvate kinase catalyzes this step. What we're going to do is we're going to transfer a phosphate group from PEP to ADP to yield an ATP molecule. Beautiful. That's the payoff phase where we are capturing energy in the form of NADH and ATP. Before we move forward, let's summarize the entire pathway and look at what we started with and what we've produced. All right, we used 1 glucose molecule, 2 ATP, and 2 NAD+. And we made 2 pyruvate, 2 NADH, and 4 ATP. But our net ATP yield per 1 glucose molecule is 2 because we invested 2 ATP in the preparatory phase. All right, let's go back to our overview diagram and put this in. We started with 1 glucose molecule and produced 2 pyruvate molecules, and we transferred electrons to NADH, which will donate these electrons and carry these electrons to the electron transport chain. We're going to deal with these NADH molecules later on, okay, in the third stage. So now, what do we do? Where do we go from here? What do we do with pyruvate? Well, glycolysis can occur aerobically with oxygen or anaerobically. When oxygen is unavailable, fermentation occurs. This is when energy is produced, ATP, but oxygen is not being used. We're going to come back to this though at the end because it's going to make so much more sense and it'll be a walk in the park. So let's just focus on aerobic respiration when oxygen is present. We mentioned before that glycolysis occurs in the cytosol. So the pyruvate molecules that we've produced needs to be transported into the mitochondrial matrix by the mitochondrial pyruvate carrier. In the mitochondria, pyruvate is converted to acetyl-CoA, which is catalyzed by the pyruvate dehydrogenase complex. In this reaction, we're going to release carbon dioxide and we're going to store energy in the form of NADH, okay? We've also produced NADH. And then we're going to add coenzyme A to form acetyl-CoA. The acetyl-CoA can then enter the citric acid cycle to be oxidized, okay? Let's break this down. This occurs, like we mentioned before, this occurs in the mitochondrial matrix, and the citric acid cycle is also a major source of NADH and FADH2, which are, again, important electron carriers that will transport their electrons to the electron transport chain. We're going to start with acetyl-CoA, which condenses with oxaloacetate to produce citrate. This is catalyzed by citrate synthase. Citrate is then transformed into isocitrate via the formation of cis-aconitate. This is done by aconitase, and this reaction is reversible. What we're doing here is citrate is first transformed into cis-aconitate via a dehydration reaction, and then turned into isocitrate by a hydration reaction. We're removing water, and then we're adding water. The hydroxyl group is moved from carbon 2 to carbon 3. Isocitrate is then oxidized to alpha-ketoglutarate, catalyzed by isocitrate dehydrogenase. This is a decarboxylation step, so we're going to be releasing carbon dioxide because isocitrate loses 1 carbon. Again, when you hear dehydrogenase, it should indicate to you that NAD+ is involved. We're going to produce NADH. There is a hydride transfer to NAD+ or NADP+, and this depends on the isozyme. It's specific for either NADP+, which is systolic, or NAD+, which is mitochondrial. That's just something to keep in mind. The next step is another oxidative decarboxylation. Alpha-ketoglutarate is oxidized to form succinyl-CoA, and we're also producing carbon dioxide. And this is catalyzed by the alpha-ketoglutarate dehydrogenase complex. NAD+ is reduced to NADH, so NAD+ accepts these electrons and will carry them to the electron transport chain. From here, Succinyl-CoA is converted to succinate, catalyzed by succinyl-CoA synthase. We're also producing GTP as a result of substrate level phosphorylation. Now, I want to quickly mention that ATP and GTP are energetically equivalent. Alright, succinate is then oxidized to fumarate in the next step. This is a dehydrogenation reaction, and we're going to be producing FADH2. This is done by succinate dehydrogenase that is actually bound to the inner mitochondrial membrane and is also conveniently attached to FAD. You'll see this awesome connection later. FADH2 can then enter the electron transport chain here, which you'll see. Fumarate is then converted to malate, catalyzed by fumarase. This is a hydration reaction right here. And then malate is oxidized to oxaloacetate by malate dehydrogenase, and NAD+ is reduced to NADH. Okay, then from here, oxaloacetate can condense with acetyl-CoA to form citrate. And we have just completed one turn of the citric acid cycle. But remember, we produce 2 pyruvate molecules in glycolysis, which means we would have 2 acetyl-CoA to begin with, or 2 acetyl-CoA entering the citric acid cycle, so it'll turn twice. That's the citric acid cycle. There are a lot of intermediates involved, so the way I personally remember these, and you can use this too, is by saying the CIA sings country songs for me on stage. Okay? Now let's summarize everything and determine our net result before moving forward. We started with acetyl-CoA. Acetyl-CoA entered the cycle and we produced 2 carbon dioxide molecules. Electrons are transferred to NADH and FADH2. One cycle of the citric acid produces 3 NADH and 1 FADH2, and these electrons are transported to the electron transport chain. We also produced 1 GTP in the substrate level phosphorylation step, which can be converted to ATP. GTP and ATP, again, are energetically equivalent. For one cycle of the citric acid cycle, 3 NADH is generated and 1 FADH2. But the citric acid cycle goes through 2 cycles, which is how we generate 6 NADH and 2 FADH2. Okay? We'll calculate the total yield per glucose later on. Let's go back to our overview diagram and put this in. 2 NADH was produced from the oxidation of pyruvate, and we produce 6 NADH and 2 FADH2 from the citric acid cycle because it'll turn twice. Moving forward, where do we go from here? Well, from glycolysis and the citric acid cycle, we've only produced 4 ATP, 2 from glycolysis and 2 ATP from the citric acid cycle. And our whole mission, our objective here is to produce ATP. But we've only made a small amount, and so to produce a lot more, this is where NADH and FADH2 come in. This is where the oxidative phosphorylation process comes into play. And we can divide oxidative phosphorylation into 2 components, the electron transport chain and chemiosmosis. Let's break down the electron transport chain. The electron transport chain consists of 4 electron carrier complexes found in the inner mitochondrial membrane. These are complex 1, which is also known as NADH dehydrogenase, complex 2, succinate dehydrogenase, complex 3, ubiquinone cytochrome C oxidoreductase, complex 4, cytochrome oxidase, and ATP synthase here, which is sometimes referred to as complex 5. Let's take a look at NADH first. What are we going to do with NADH? NADH transfers its electrons to complex 1 and forms NAD+. These electrons get passed on from one carrier to the next within complex 1 and then transfers them to ubiquinone or coenzyme Q. So then coenzyme Q is reduced to QH2 or ubiquinol. Then QH2 or ubiquinol will pass the electrons to complex 3. QH2 is mobile. It's diffused in the lipid bilayer, so it's going to carry these electrons to complex 3. Now, when electrons are passed along the transport chain, there is a release of free energy, and this energy is used to pump protons from the matrix to the intermembrane space. This proton pump is caused by the energy of the electron transfer, because remember what we said, the more reduced a molecule is, the more energy it has, and because NADH transferred the electrons to complex 1, it's high in energy, and we're going to use that energy to pump protons from the matrix to the intermembrane space. Okay, specifically, we're going to pump 4 protons from the matrix to the intermembrane space. And this is how the proton gradient is established. We have hydrogen ions in the intermembrane space. It'll make more sense in a minute. So NADH donates its electrons to complex 1. Moving forward with FADH2, what do we do with FADH2? FADH2 donates its electrons to complex 2 and becomes FAD. It only donates it to complex 2 and complex 2 alone. Complex 2 is called succinate dehydrogenase, and if we refer back to the citric acid cycle, it is a membrane-bound enzyme. It's right here. This is the connection that I was talking about earlier. Gorgeous. And then the electrons are passed to CoQ, which is then reduced to ubiquinol. Again, QH2 is mobile, so it can carry these electrons and move through the membrane and donate the electrons to the next complex, complex 3. Now, in complex 2, protons are not being pumped from the matrix to the intermembrane space, and so the QH2 ubiquinol will pass the electrons to complex 3. Okay, now we're at complex 3. Let's slow down for a second. Complex 3 is a multi-subunit complex that contains cytochrome B, C1, and iron-sulfur centers. So QH2 will donate the electrons to complex 3 and then be oxidized back to coenzyme Q. And complex 3 will pass the electrons along the carriers that make up complex 3 until it passes the electrons to cytochrome C, this right here. And for complex 3, we're going to pump protons into the intermembrane space right here. So there is a release of energy that is used to pump the protons from the matrix into the intermembrane space. Now we're at cytochrome C. Cytochrome C is a soluble protein. It's a mobile carrier, and so it's able to move in the intermembrane space to complex 4 and drop off the electrons. And the electrons, what's going to happen, the electrons are going to be transferred along the different carrier molecules that make up complex 4 until it reaches the final electron acceptor, oxygen, molecular oxygen, and we produce water. So complex 4 catalyzes the formation of water right here. And we're also going to pump 2 protons into the intermembrane space. Again, the pumping of these protons from the matrix to the intermembrane space is establishing and creating that proton gradient because the intermembrane space is more positively charged compared to the matrix due to the protons, the positive boys, leaving the matrix. Now, this proton gradient is going to produce what's called the proton motive force, which is just the potential energy from the proton concentration difference, because remember, we're pumping protons from the matrix to the intermembrane space, so there's a proton concentration difference. Then the proton motive force is going to activate this ATP synthase complex and synthesize ATP. Let's go through this. ATP synthase has 2 components. The F0, which is located across the membrane, okay, this right here. This is the section that provides the proton channel. Proteins are going to flow through here from an area of high concentration to an area of low concentration. And the second component is the F1. This is where ATP synthesis occurs, but both of these components work together and they use the proton gradient. Combining the 2 parts, what does ATP synthase do? Let's go through this. It allows protons to flow back into the matrix to stimulate the production of ATP. ADP and inorganic phosphate combine to make ATP. The proton gradient, or the electrical gradient, has potential energy. We've already established that. And that energy is going to be used and converted into the chemical energy of ATP. This process here is called chemiosmosis. We're pushing protons from an area of high concentration to an area of low concentration. The proton concentration difference between the intermembrane space and the matrix holds potential energy, the energy that was produced from transferring electrons. And as protons move from high concentration to low concentration through ATP synthase, this stimulates the production of ATP. And if we take the electron transport chain, okay, and chemiosmosis, this is called oxidative phosphorylation. The electron transfer to oxygen is linked or coupled to ATP synthesis. Wow, look at that. How beautiful is that? That was quite intense, actually. So let's take everything we've covered, let's step back and look at the overview picture, and let's determine the total ATP yield per glucose molecule. First of all, what's our mission? What are we doing here? What are we trying to achieve? We're trying to produce ATP. We're trying to generate energy. We're harvesting energy. We're harvesting the energy of glucose for ATP synthesis. Again, there are 3 stages: glycolysis, pyruvate oxidation, and the citric acid cycle, and oxidative phosphorylation. Let's start with glycolysis. So in glycolysis, we produce net 2 ATP and 2 NADH. Now, you can make 2.5 ATP for each NADH molecule and 1.5 ATP for each FADH2 molecule. So for this NADH, the ATP equivalent gives us 5. For glycolysis, our total here is 7. Then moving on, the pyruvate molecules we've produced are oxidized to acetyl-CoA. We generated 2 NADH, so that gives us 5 ATP. Then the acetyl-CoA enters the citric acid cycle. Let's calculate this. 2 ATP was generated from substrate level phosphorylation, and then 6 NADH was produced, and we multiply that, 2.5, and then 2 FADH2 was produced, we multiply that by 1.5. Recall that for one cycle of the citric acid cycle, 3 NADH are generated and 1 FADH2 is generated. But the citric acid cycle goes through 2 turns, 2 cycles, which is how we generate the 6 NADH and 2 FADH2. Taking all of this into account, our total number of ATP molecules produced per 1 glucose molecule that is oxidized under aerobic conditions with oxygen present can produce a total of 32 ATP. So the total yield per glucose is 32. Absolutely beautiful. How lovely is it to see the overview picture and actually understand what's going on? A walk in the park. That's cellular respiration. Before we end this lecture, let's briefly go through what happens to pyruvate under anaerobic conditions without oxygen present. Okay, let's slow down for a second. Referring back to the glycolysis pathway, we produced NADH during the payoff phase, okay? This NADH, like we've covered, will then be reoxidized to NAD+ when it donates its electron to the electron transport chain, to complex 1. So then, what happens when you are in a hypoxic condition, when oxygen is not available? Well, the NADH that we've produced in glycolysis can't be transported to the respiratory chain, to the electron transport chain, where it can be reoxidized by oxygen. And if we can't reoxidize NADH, then there will be insufficient NAD+ to be the electron acceptor, the electron carrier, when glyceraldehyde 3-phosphate is oxidized, and that would inhibit glycolysis. Glycolysis won't continue. So our goal here is we need to reproduce NAD+ in another way, and that is where fermentation comes in. Fermentation occurs when energy is harvested, ATP, but oxygen is not being used, and the concentration of NAD+ or NADH remains unchanged. Okay, there are 2 types of fermentation involving pyruvate, lactic, and ethanol fermentation, but let's just go through lactic fermentation in this video. If you'd like to see ethanol fermentation, you can check out the Pyruvate Metabolism lecture. We covered both types. Lactic fermentation happens when our muscles are working hard. So for example, when you're sprinting, okay, you're going at it. Our goal here, again, is to regenerate NAD+. So pyruvate is converted to lactate, but the enzyme lactate dehydrogenase, remember, when you hear dehydrogenase, NAD plus is involved. We're producing NAD plus, and there's going to be no net change in the number of NAD plus and NADH because 2 pyruvate molecules are reduced to 2 lactate molecules. And going back to glyceraldehyde 3-phosphate, okay, right here, it turns 2 molecules of NAD plus into 2 molecules of NADH. So the concentration of NAD plus and NADH remains unchanged. All right. We produce lactate. What do we do with lactate? Where is lactate going to go? Well, lactate can be recycled. We can transport lactate to the liver, where it can be converted back to glucose via gluconeogenesis. Gluconeogenesis is the production of glucose from non-carbohydrate sources. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!